In a healthy ecosystem, we have lots of biodiversity, lots of different species, and they all perform different roles. So we need to know what the species are that are in the system so we can understand it and then put protections around those systems. We left Wellington so that we could head out towards the Bounty Trough to start an expedition to discover new species. The Bounty Trough was chosen as our area of investigation because we plotted some data on the map of all the places that we had biological samples from NIWA and Te Papa collections, and we found that there was a big gap in the Bounty Trough, so it was really exciting to be able to choose an area that we've never been to before. When we go to a new site, we use multi-beam to take a map of the seafloor, and once we have a map, we start to plan where we're going to go.
Probably tailless from this cliff, so there might
We want to use our video camera, the deep-towed imaging system, to get our eyes on what the seafloor looks like.
Okay, bridge, going over the side now. Yeah, definitely going down into the channel now.
So we just completed a detus, which went down to 4,160 metres deep, which is really amazing. It's the first time detus has ever gone that deep. Yeah, we had a bit of a celebration in the lab when that happened. If it's muddy, we're going to use a particular type of equipment. We can take a core of the mud with a multi-corer, or we can use a seamount sled or a beam trawl, depending on whether it's rocky with the seamount sled or muddy, to take a sample of the animals that live just on top of the mud. When a sample first comes on deck, the scientists are all really excited, so we all kind of swarm in on top of the sample and start picking out all of the precious animals.
Rocking it!
got a whole lot of invertebrates, mollusks, echinoderms, crustaceans, polychaete worms, sea anemones, some live corals, some hydroids. So the list goes on. There's a lot in there.
I'm not really sure what this is. It's a bit hard to know at this stage. Very exciting. Yeah. There's a lot of sample sorting initially, and then the samples get taken into the container where they get labelled and assigned a unique catalogue number. So that catalogue number follows them through into our collections database, and then we can use that to track them through the whole system. So now we're steaming back towards Wellington, unload the ship and take all those samples back to NIWA and Te Papa and get them ready for the experts eagerly waiting for our arrival back in Wellington.
Once we have a sample, we look at the animals, divide them into the families, and then you whittle it down to what genus, what species. You compare it to all the literature, you look at it, you draw it. We're also sending off samples for sequencing so that we can look at a molecular level.
NIWA invertebrate collection, where most of the invertebrates will be going. That collection's a nationally significant collection, so it's important to keep that database and that collection accessible to as many people as possible in future when it's sitting on the shelf in the collection, and then we might be able to loan that out to future researchers for a whole variety of different studies, things we haven't even thought about yet. We're keeping these specimens for a really long time in our collections to make them accessible to future researchers.
Be able to understand the diversity of life and to be able to describe a new species, we've got to compare it with the other animals which have already been described, and you can't do that without collections.
We want to understand what the ecosystem is and what the biodiversity that lives in it is, so that we can understand what the pressures are. Understanding what they are is the baseline fundamental information that we need to know. We need to understand biodiversity so we know what we need to protect.